Tissue analysis of the vocal folds' cellular and biochemical aspects. We'll look at two promising ways to analyze vocal fold tissue, especially during real-life movement. First, voice-related biomarkers assessed using validated tools like acoustic measures, maximum phonation time, the voice handicap index, and the Grabe scale. These offer a clearer view of vocal function. Second, optical coherence tomography, often called an optical biopsy. It visualizes tissue and detects abnormalities without removal. It's useful for studying hormonal and biochemical properties in live conditions. The vocal folds are our primary tone generator. This study highlights new ways to assess their function using biomarkers based on UEP and ELS consensus. Key tools: voice handicap index, Grabe scale, and acoustic measures like frequency, jitter, shimmer, and harmonics to noise. Maximum phonation time also offers valuable insights into vocal performance. This slide summarizes UEP's four main tools: voice handicap index, the patient's own view of voice issues; Grabe scale, clinician's perceptual assessment; acoustic measures, frequency, jitter, shimmer, harmonics to noise; maximum phonation time reveals vocal and respiratory efficiency. These cover patient feedback, expert validation, objective data, and psychological airflow metrics. Though we understand elements like elastin and fibrinogen, clinical application remains limited, especially for complex syndromes or neurodegenerative disorders. Biopsy risks altering tissue behavior, which is why optical coherence tomography is vital, allowing in vivo testing without interference. We lack biochemical insight into how treatments affect tissue-specific abnormalities, particularly involving genetic or hormonal imbalances. We also need baseline data on healthy vocal fold movement to better understand these effects. The article calls for an interdisciplinary approach, combining biochemical research with clinical tools like biomarkers and optical coherence tomography. This integration is crucial for improving diagnosis and treatment of disorders influenced by hormonal, genetic, neurodegenerative, or environmental factors. Genetics help us understand pitch and its biochemical ties, though much is still unknown. Voice pitch and vowel acoustics are heritable. One study linked pitch to gene variant ABCC9, showing genetic association. Hormones, especially during puberty, strongly affect vocal fold development. Research is fragmented, but estrogen and testosterone are clearly linked to voice changes in both males and females, with predictive value over time. Promising research has looked at normal vocal fold structure, cells, glands, elastin, fibrinogen, and the extracellular matrix. Still, clinical applications remain rare. One study on oral mucosa showed how cells differentiate during function, insights that may inform future vocal fold research. Some findings offer hope. In young patients with motor deficiencies, vocal fold regulation remains partially intact. Training can improve function as shown through voice range profile analysis, suggesting rehabilitation potential. Environmental factors like radiation affect voice, yet remain understudied. A study on children exposed to the Chernobyl disaster showed reduced vocal fold movement and pitch range during puberty, highlighting the need to understand environmental impacts. OCT allows real-time, non-invasive tissue imaging showing capillarities, glands, swelling, and even infection-related cells. It offers precise abnormalities detection, enhancing clinical analysis. New tools like voice-related biomarkers and OCT are pushing the field forward. OCT helps define and measure vocal fold biochemical abnormalities under specific conditions, improving diagnosis and treatment, and aiding future research comparisons. Thank you for listening.